Let me just give you an overview to begin with, and I have to wrestle with the technology here a little bit of what I'm going to talk about. First, I'll just tell you a little bit about what the Sunshine Project is, so it's clear sort of where I'm coming from and what I do. Then for the presentation, we will talk about what's happened since 2001 in the U.S. biodefense budget and how that is played out across the country in academic and other types of laboratories. Talk a little bit about what high containment laboratories are, sort of what the physical facilities are that we're talking about, that the focus is on. Talk a little bit about how they've proliferated. And then turn our sights to well, for lack of a better word, legal system. It's really sort of a quasi-legal guidelines-based system for oversight of those facilities. And then try to step back before concluding a little bit and put this tremendous proliferation of biodefense research that's happened in the United States in the last 6 or 7 years into a sort of broader, both scientific and kind of geopolitical context before coming to some conclusions, which I'm afraid aren't terribly optimistic at this point in time, but you know, things can change fast. So, moving on, just to tell you a little bit about the Sunshine Project, so you know sort of where I'm coming from. We were founded at the end of '99. Basically, myself and a German colleague in Hamburg, both of us had worked in some of the sort of biotech battles of the '90s over genetic engineering and agriculture. My background was primarily in working on intellectual property issues because, of course, patents were a relatively new thing when it came to living organisms and parts thereof. Both of us having worked in agriculture for a long time and having read a lot of biotech patents and other documents kind of became concerned about what you could do if you really set out to do something bad with a lot of the technology that was coming into broader use in the mid to late 90s. And so we decided to start an NGO, which really is kind of the only one of its kind that's not academic based or sort of DC based to work on biological weapons issues. No government money, obviously. If you know the organization at all, you know they'd really rather put us out of business than give us any grants. So it's foundation and donor supported. You're looking at the full-time staff, so that's it. There's various other people that work in one capacity or another, but I'm the only person currently who's working full-time for the project. The main things that I do are track the U.S. biodefense program and work on related biological weapons convention issues. That's the BWC. I also do a lot of work in non-lethal weapons, biochemical non-lethal weapons. I'm not afraid that the United States is going to start, weaponizing anthrax again and dropping anthrax bombs on anybody, but I am a little bit worried about some biochemicals and their use in things like riot control. There is significant military interest in that area, so that's one thing that I work on. And recently I've taken the plunge to start working on pandemic influenza issues, which maybe we'll get into a little bit later on in the presentation. And since I was talking to lawyers, I thought I would mention that I actually do a little bit of litigation. One of the things that I do to track the biodefense program is do a huge amount of open records work, federal FOIA, but also state open records requests. Have two, I think about to be a third suit going under the Texas Public Information Act. So I've often thought if I didn't do biological weapons, I would do freedom of information work, because I think it's just, it's so incredibly important. It's so key to what I do. And as Stephen mentioned, there's the website. And not only can you see all the wonderful things that we do at the website, but you can contribute too if you just happen to get the urge while you're there. So with that as sort of an introduction to who I am and what I do, let's move on to sort of the meat of things. The real sort of backstory to where the current boom in biodefense research begins is, of course, with 911 and the anthrax letters. I'm not going to reach that far back or really talk about that, but I think how that led us sort of as a society to spend a lot more time worrying about security and worrying in particular about biological and chemical and WMD security is pretty clear to everybody. But what happened in a practical sense was that after 2001, there was a tremendous spike in federal money that was made available to study biological weapons agents. So the proliferations of labs and everything that you see across the country now is not the result of some sudden, spontaneous, massive scientific interest in some really weird bugs. It's driven by the money. And there's a tremendous amount of money that has gone out of the federal coffers and into building high containment labs, into funding research on anthrax, Ebola, you name it. NGOs like to do budget figures. I'm always a little bit suspicious of them, but these are the best that I'm aware of. But if you look, the growth has been pretty astronomical. You know, if fiscal 2001, the federal biodefense budget, by this estimate, was slightly over $1 billion. I've seen other estimates that are slightly under $1 billion. Now going to proposed, or the request, FY08, it's about $7 billion a year. One of the reasons why I don't like these kind of budget figures, though, is that they are based on the open budget. And we know that there are two other major kinds of biodefense money that are out there, that being in the intelligence budgets, which of course they don't even release the total figure of what we spend on intelligence in the US, and we know that there are applied biodefense programs, wet programs, working with bugs that are managed by the CIA and perhaps other agencies. And they, of course, don't include black appropriations. So if anything, this roughly seven-fold increase in the amount of spending, which excludes BioShield, incidentally. BioShield is just a subsidy program, basically, for industry. It's not R&D. So roughly this seven-fold increase in spending since 2001 is definitely an underestimate. Okay, the factor is probably more like 10, but we just don't have access to those numbers. I'm sorry?
Can you clarify the term black appropriations?
Those would be appropriations in the federal budget for programs that just have a code name, basically. You know, the whatever it is, the White Knight project. And that's all that's publicly.
Unrelated to intelligence community programs?
Yes, there are. So where do you, was that, sorry.
Defense or offense? Biodefense?
Okay, well, we're skipping ahead here, but let's talk about it. Yeah, that's the question. That's always the question, right? The intrinsic in the nature of doing research related to these bugs is that a lot of the science has both offensive and defensive applications. Like I said, I don't personally fear that the United States is going to start dropping anthrax bombs on anybody else. But the problem is that if you, for example, want to better learn the process of anthrax infection by a pulmonary route, what you have to do is you have to basically teach a lot of people how to produce, nice, finely prepared, sporulated anthrax. You basically have to weaponize the agent. And then you have to learn a lot about its aerobiology. So intrinsic in the biodefense program is that a lot of the work, should we choose to do so, or should a person involved in the program choose to do so, can be applied to do harm rather than to protect us or for some other intellectual end, which more typically it is than direct applied work on some sort of countermeasure. Does that sort of satisfy you? I mean, it's a very, I mean, this is what's called the dual use issue, and tome upon tome has been written about it, and you could go on and go talk about dual use forever. I think for our purposes here, I think the way I would prefer to sort of leave it is that although the intent may be defensive, what you have to understand is that the nature of the work generates knowledge and skills and a set of people and, to an extent, facilities that are equipped to go offensive. Or maybe another way to put it would be, on a routine basis in our biodefense program, we do activities that, were they to take place at a lab in Tehran, we would probably bomb them. Except it's okay for us to do it because we have confidence that we are not going to, bomb them with anthrax. We're going to bomb them with just high explosives. So that's the nature of the beast to an extent. Obviously, if you're going to massively expand work on these bugs, you have to have the right places and the right conditions to do it safely, so you're not going to kill yourself in the process. So the focus in terms of the expansion of the biodefense program is on one focus is on facilities. And just to be clear, there are really two kinds of facilities. There are 4 levels of laboratories, 1, 2, 3, 4, with 3 and 4 being the highest, most secure facilities. Level 1 and level 2 labs generally, in almost all cases, will not be handling a biological weapons agent. There are a few BW agents that are handled at level 2, but that's really becoming increasingly an anomaly. Level 3 labs are, in general, for things that if you became infected with them and they were not treated, they would kill you. It's life-threatening disease, but if you know that you're infected and if you have access to medical care, you're typically going to successfully survive infection. So this is anthrax, plague, tularemia. The real outlier are some of the pandemic influenzas, because you very well might die even if you are treated with those, but they're nevertheless handled at BSL3 for complicated reasons. But the characteristics of the lab would be that they'll have HEPA, the very microfine filtration of the air that moves out of the lab. The labs will be at negative pressure relative to the outside, so if this were a lab here, the air pressure would be less than it is outside those doors, so that we wouldn't blow microbes out the door. And of course, even within the lab, there'll be rather complex and specialized cabinets in which the bugs themselves are actually handled. The thing about BSL3 Labs, and this annoyed me here in Seattle, and it annoys me across the country because we've been building hundreds of new ones in the last few years, is that typically proponents of the facility will sort of say, well, BSL3 is a BSL3 is a BSL3. And that's really not the case. There are probably a couple thousand BSL3 labs in the US, and these range from very small diagnostic facilities, which are absolutely critical to public health. Okay, if you got somebody that walks in the door and they look like they might have have some sort of skin lesion that looks like it might be anthrax, if you're a doctor, if you're going to take a sample and you're going to want to handle that sample with, the maximum safety precautions that you can. So they range from very small facilities that have very immediate and real public health uses to much larger facilities that are entirely research oriented. And so a BSL3 is not a BSL3 is not a BSL3. You really have to look at the facility to look at the equipment, the size, the nature of the work that's going on inside it to sort of understand what the research agenda is and whether it primarily relates to providing medical care, for example, or it could be for research on HIV. If you have really large quantities of the AIDS virus, it requires BSL3, or if it's really for biodefense related work. BSL4s are much rarer. And there's only about five of them operational in the US, although we've got about 10 more that are on the way. In fact, we're going to probably multiply the square footage of BSL-4 facilities in the US by more than a factor of 10 by a few years from now. Historically, they were really only at Fort Detrick in Maryland, which was the home of the US offensive biological weapons program before 1969, and since has been the home of the Army defensive program and its CDC, the Centers for Disease Control in Atlanta. Now there are two more facilities operational in Texas. There's a new one both at Fort Detrick that's operational and, well, it's not quite operational yet, one that's finished and should be going hot soon at CDC, a second lab there. And there's a bunch more on the way. And these tend to be, well, almost always there's a lot of public focus on the BSL-4 facilities. This is where, if you read Richard Preston books or something like that, this is the hot zone. This is where the scientists wear the funky space suits and really thick, about 18 layers of gloves, and even those gloves reach inside of other gloves that reach inside the cabinet. This is where the bugs, and there are really not that very many of them, but this is where the very unusual rare bugs, generally viral hemorrhagic fevers like Ebola, Marburg, are handled, where if you get infected, you're pretty much going to die. They're very rare. Survivors are very rare from being infected with these types of bugs. So that's these are the two major types of facilities. Just two additions to that. There are animal designations for both of these. So there's animal BSL-3 and animal BSL-4. And so sometimes you'll see ABSL-3, ABSL-4. And pretty worrying, actually, to me, and I think to a lot of other people, is that in part because of scientific competition and because of the relative scarcity of BSL-4 labs, and because we're putting so many people behind the driver's seat of these very dangerous bugs who, well, they simply weren't that many people doing it only 7 or 8 years ago. There's a lot of BSL-3 plus and kind of improvised BSL-3 labs with additional sort of shower out or additional types of features that typically would be related to a BSL-4 lab. And in fact, at UW is one of the places where one of these facilities exists. It's kind of worrying from a biosafety standpoint because they're really pushing the envelope with some of this stuff. The level 4 labs are level 4 labs for a reason. They were designed with maximum engineering controls to do, the engineering controls are never going to be perfect because they're never going to perfect human beings, but, that level exists for a reason, and increasingly you see more and more kind of risky work taking place in BSL-3s. The most risky, in my opinion, of that is the work with pandemic flu. But perhaps we'll have a chance to get to more of that in a little bit. This map is a little bit dated. It's the last time I updated it was more than a year ago, but yeah, it's a bit blurry. But just to give you a sense of these are not, like I said, there's probably 2,000 BSL-3s in the U.S. So these are not a map of the BSL-3s in the U.S. or the BSL-4s. These are the major new biodefense facilities that are being built. So it's the scale of expansion of the program it's completely unprecedented. The only real precedent that I can think of that exists would be the Soviet Union when it went offensive in the 80s. Built out facilities of a somewhat different kind because they were production facilities, but at a scale like this. The historical centers were Fort Detrick in Maryland, and to an extent Edgewood Arsenal, which is also sort of north of Baltimore, and Dugway Proving Ground in Utah. And those continue to be major facilities to this day, but through grants from NIH, from Homeland Security, from the Department of Energy. Now these labs are cropping up everywhere. The sort of old nuclear friends, Lawrence Livermore Lab in California, Los Alamos in New Mexico, have built BSL3s in the last few years because this is kind of a growth industry for government-funded security-related science. So in total now, according to the latest information out of CDC, which is like squeezing water from a rock, they don't tell anybody about anything that's going on in what's called the Select Agent Program. We have now about 413 was the last that I heard labs in the United States handling biological weapons agents, and more than 15,000 people registered. So they're really all over the country. And in the next slide, you're going to see that a huge number of the people that are involved in this research have no prior experience handling these agents. The federal reply, how are we going to handle safety? It's generally acknowledged, and it was generally acknowledged 2003, 2004, 2005, that you don't go from having, for example, as a rough figure, 8 or 9 active programs studying anthrax to having now probably over 200 in the United States overnight without creating all sorts of risks. So it's generally acknowledged that this huge expansion has created risks of a few types. There's the risk of an accident, a bug leaking out the window, so to speak. There's also the risk of an insider, being a person that works in a facility who, for whatever reason, goes nuts or has a vendetta, chooses to apply their skills to cause harm. There's evidence that there's circumstances suggest and evidence suggests that that's what happened with the anthrax letters in 2001. Or in some scenarios, if you are concerned that foreign terrorists intend to commit acts of bioterrorism in the United States, probably the easiest way to do it is not to bring in a vial of plague on the airliner, but it's to acquire it from a U.S. Biodefense Lab and figure out and acquire the means to use it. So there are a series of risks that are posed by the expansion of these facilities. Now, the federal reply, as how the Bush administration its reply about how we're going to handle the risks has been twofold. One is to say that review of this dual-use research, right? There's concern that maybe scientists might not really think through what they're doing very clearly, and they might, start going down the wrong path in their lab, and maybe they're going to do stuff that really has so much offensive application and isn't really justifiable for defensive purposes. Federal reply is, in order to contain that risk, what we're going to do is we're going to hand this job of oversight to local committees called institutional biosafety committees. And on the other hand, there's a law, there's the Bioterrorism Act of 2002, establishes that people, in order to handle these agents, need to be registered. And the facilities themselves are registered. So CDC has a registry of all these places, which is the only reason why we know there's 413 labs, roughly, and something over 15,000 people. There are, for labs, also additional rules and quasi-rules that apply. These are the main biosafety manual is called the BMBL, published by CDC. OSHA has rules. There's a bloodborne pathogen standard. The U.S. Army has its own biosafety program, but the system is really fractured. And to talk about it, I'm just going to take a couple of examples. I could talk about institutional biosafety committees all night long if you let me. It's been a major focus of mine for the last, since 2003. These are really the committees. So to oversee this system, which is now 10 times as big as it used to be, we've got roughly, 20 times as many people working, for example, with anthrax in labs in the United States as we had, only 7 or 8 years ago. To oversee this system, the federal response is these institutional biosafety committees are going to do it. Well, first of all, the federal government hasn't even actually told institutional biosafety committees what it is exactly that they're supposed to do. But even sort of more worrying than that is they don't operate under any federal regulation, okay? They operate under guidelines because we decided back in the 1970s when genetic engineering was young that we were not going to regulate laboratory biosafety in the United States in effect. And so what happened was a system called the NIH guidelines, federal guidelines called the NIH guidelines emerged to regulate or to quasi-regulate experiments with recombinant DNA, so biotechnology experiments. Okay, so I won't get into IBCs too much. In effect, what I've found, and I think what some other people that are looking into it now have found, is that these committees are allegedly required at all institutions that have NIH funding for biotechnology research. Under USDA, oddly enough, although NIH doesn't have a regulation, for USDA and DOE there are actually regulations that they have to comply with the NIH rules that are not regulatory, and under NIH it's bizarre. These committees basically in many cases don't meet. They don't even exist. When they do exist, they may consist of just a list of names. To give you just a few examples, I filed a series of requests. The NIH guidelines have public access provisions. The committees are supposed to make their minutes available to the public on request. I asked all the committees in the country for their minutes. And I got things back like the minutes of the first ever IBC meeting at Utah State University, where they provided a list to the committee of the 40 or 50 projects that the committee had already approved in the last 5 or 6 years before it had ever met. And this kind of thing happened all over the country. So you have the core local oversight system, this IBC, that in many, many places across the country simply doesn't perform its task. And then we'll get to the select agent rule in a minute. All right, coupled with this has been as it is across the board with this administration really, a tremendous increase in the application of national security or classification of records. We don't know how much classified biodefense research is out there. We know that there's a lot of it. I run into it. You can sort of, using the Freedom of Information Act, you can occasionally, sort of accidentally bump into projects like this. So how much is out there? We don't know. I mean, only history will really tell us. But perhaps, even more worrying than that has been the huge expansion of the application of these sort of quasi-secrets, of sensitive but unclassified stamps and monikers on information about federally sponsored research. I put this one page, which obviously you can't read up here because it's an example that really aggravates me. This is a CDC document, stamped SBU here, sensitive but unclassified, which I got under Texas law. And what it's saying is that Texas A&M University, which is in very deep doo-doo with the federal government right now, at the top it's saying permitted people unauthorized people access to these agents. It permitted people, it follows, the next sort of violation of the CFR says that it allowed people with no training to handle these agents to handle the agents. And then at the end, the last violation of the CFR is that one of the principal researchers there was conducting unauthorized genetic engineering experiments with brucella. And were it not for the relative power of the Texas Act, I was able to get this. But basically, we know that this is one page of a 22-page letter that outlined probably 15, it depends on how you count, 15 or 20 violations of federal regulation in Texas A&M labs. We know that there are probably about another 120 reports like this that exist at CDC of the ones that they've actually investigated, and you're not going to get anything from any of them. So and it's not classified. Okay, none of this information is classified. We know that there are deep problems with the system, that oversight isn't working, but you're not going to get it out of them because basically they stamp everything SBU. And if you file a FOIA with the Centers for Disease Control, which is supposed to be overseeing this work under the select agent rule, they don't even conduct a search. They just wham, they just deny it. They say that if we had it, it would be exempt. Or they send what's called a GLOMAR response, which is basically to refuse to confirm or deny the existence of the records. But we know through the Dingle Committee, which actually was able to exert more pressure than your average public requester on them, that there have been more than 100 investigations and reports of accidental releases of select agents in the United States since 2001. What this has engendered at the sort of local level is a lot of really bizarre behavior by sort of our institutions, by universities like this one and many others across the country, that all they know and all they really understand is that the federal government is really uptight about this biological weapons agent research. And so you ask them for information and they do really bizarre things. And I mean, some of the things that, well, I've been reported to the FBI many times as a potential bioterrorist because I was asking about this information. But they will. As an example, I had one university in South Carolina. They had a research project on the type of herpes virus that causes cold sores. And they redacted all the information about that because it was a biological agent. So it has really insidious effects on sort of the transparency of our institutions and well, it's very ironic if one considers the dearness with which the biological science community holds its academic freedom. That now, if you ask for this type of information, it's incredibly difficult at times to get information about research on biological agents that aren't even really of. To try to put it all in context before giving you some conclusions, I just wanted to go through a few numbers here. These are a little bit dated, but to sort of say, okay, we've increased our spending on research on biological weapons agents by about a factor of 10. Okay, how does this compare? How do we compare this? How do we understand this? And one way to do it is to look at how it compares with research on other diseases and what are the diseases that people actually die of, infectious diseases. So these are FY. This is a chart that compares fiscal 1999 through 2001 spending on certain diseases versus fiscal 2002 through 2004 spending. And what you can see is that the spending on the biological weapons, bacteria, and viruses has gone up 2,300%, almost 2,000%, from the viruses from about $6 million in total, 99 through 2001. This is at the National Institute of Allergy and Infectious Disease, to 120 million from 2002 to 2004. Meanwhile, these numbers down here, this is funding for basic research. Okay, so this is funding on like model systems. This is the sort of bread and butter science funding. You have declines in the amount of money that's coming out of the National Institutes of Allergy and Infectious Disease to study these. That's really not very legible, is it? Okay. Well, I'll just read the figures to you. I apologize for the technological problems. Another way to look at it would be to compare the U.S. deaths. What do people die of versus what are we investing in terms of spending. And if you look at the 6 bacteria that are the priority biological weapons agents, which are tularemia, brucella, plague, anthrax, and 2 others. Basically, the likelihood of a person being diagnosed with any of those 6 in 2003 was about 1 in 1,230,000. So it was less than 1 in a million Americans actually had one of these infections. Whereas if you look at 6 other infectious diseases like gonorrhea, AIDS, syphilis, there's about 1 in 600. Plague, for example, we just had a guy, and I believe it was in Arizona, die of plague. That's about the annual death rate in the United States of plague, about 1 person. It was the same in 2003. There was 1 person that died. Whereas if you look at, the total figures from the 6 BW agents, you had 234 people died in 2003, or excuse me, 230 people were diagnosed. By comparison, there were 400 kids that were born with syphilis, which is an entirely preventable thing, right? So we've massively increased our funding for these diseases that basically Americans don't die of, and while our public health system continues to deteriorate, as evidenced by things like children being born with syphilis. All right, so looking internationally and looking at the science, while we're busily expanding the biodefense program, biotechnology is really going global. So in other words, it's no longer us that are really producing biotech products anymore. There's massive expansion in Asia in particular, in Southeast Asia, in China, in South Asia. So this proliferation of biotech has got a lot of security type people in the US government and in other governments, a little bit worried. And at the same time, in addition to, which I don't really have time to get into, your sort of standard advances in genetic engineering, which continues to move ahead at a very, rapid rate. Another emerging issue from a technological front that's coming in is synthetic biology, because now it's really, it's not easy yet, but you can order pandemic flu in the mail. That's a bit of an exaggeration. It's not quite that easy. A virus like SARS, which is a pretty big virus, it's more than 30,000 base pairs long, has been synthesized from scratch. So there's a lot of concern that, from a security standpoint, that the emergence of synthetic biology, in particular the sort of gene foundries, are going to facilitate access to these agents to a wide variety of people who might not be the people that we want handling them. Another good example on the synthetic front, 1918 flu. It didn't exist. It was dead. It was gone. Now it exists as a real live bug. It was basically put back together through synthesis. So if we're worried to death about the sort of black potential of biotech, one way to try to allay our fears is to see what we can do in terms of cooperation internationally. And the logical people that would be able to help us with this, so that we can, instead of being worried about what the Chinese are doing in their BSL3 labs, which is what our guys at the Pentagon are worrying about, maybe we can come up with some sort of methods of cooperation that are gonna work. And unfortunately, it's not going very well at this point. The Biological Weapons Convention, which is the international treaty which prohibits biological warfare, is basically an honor agreement. It was concluded during the Cold War. There's no verification mechanism. There was almost a verification mechanism. It was killed by the Bush administration in 2001. So we basically have a Biological Weapons Convention that's sitting around talking about non-binding things. There's no move forward there to, for example, improve our confidence in what's going on in labs in other countries by creating an international inspectorate. In fact, we did have an international biological weapons inspectorate. It was UNMAVIC. It was founded for Iraq, even though we didn't let them back into Iraq. It was just disbanded. So we've, unfortunately, at the international level, lost a set of expertise that we had. And logically, WHO might be, the World Health Organization might be another organization that would help us, but unfortunately and fortunately, there's tremendous resistance in the medical community to the sort of securitization of the work of WHO, and it's really for very understandable reasons. I'll have to leave it at that since I'm running out of time. Maybe we can talk about that if there's people interested in it in the questions. So to conclude, I think that undoubtedly, if you look at the biodefense program in broadest perspective, we have unfortunately entered into a phase where we are basically in an extended period of plumbing the depths of the sort of dark potential of biotech. The question is, where are we going to bottom out? The Department of Homeland Security, for example, is enthusiastic about types of biodefense studies called red teaming, which is a hangover term from the Cold War. What it means is that you set loose a bunch of guys to pretend that they're the terrorists or pretend that they're another country hostile to the United States, and then they will literally go about in the lab, perhaps, or out in the desert in Nevada, acting as if they were terrorists. And this raises some treaty issues, but it's very unclear where we're going to bottom out in this, and we have diverted a tremendous amount of resources away from research on public health and research on the things that Americans actually die of. There are, unfortunately I only have problems for conclusions, I'm afraid. The biological sciences are being, particularly at universities, are being tremendously impacted, although they don't, and they're not coming to terms with it yet, with all of this security money. Basically, until recently, people didn't get national security funding in the biology department, and now that's happening everywhere. And the biology professors would like to think that that money will continue to be available, and there are going to be no strings attached to it. But there are strings. Right now, the Department of Homeland Security is signing, secret or quasi-secret provisions on research contracts to universities that say that your research results have to be sent to Washington and looked at by the Department of Homeland Security before you can release them to anybody else. That kind of thing is happening on campuses, and the biosciences community has yet to come to terms with the long-term impact of being linked to the national security apparatus in the way that it is. Our regulatory system, to the extent that it's actually regulatory and not guidelines or manual based, is a complete disaster, and it's fractured. And this is one of the things that they're looking at in Washington in the House Energy and Commerce Committee. And finally, I think that there's, I hope that, and this may be the point that's perhaps a bit optimistic, is that, perhaps we're reaching the point where we're going to emerge a little bit from our collective paranoia that set in 2002, 2003, where everyone was basically so terrified of biological weapons and biological warfare that we somehow intertwined our notions of public health and biodefense in a way such that we can't distinguish between what serves the public health good and what will prevent people from illness or disease, and what Americans really die of, and research on a disease like glanders, which no American has contracted since the 1940s, but which we're nevertheless pumping more and more money into studying. So hopefully, I think that now that we're 7 or 8 years beyond that, or 6 or 7 years beyond that, that perhaps we're reaching a point where we can have a more rational dialogue, sort of nationally, and public dialogue about whether this incredible proliferation of very expensive facilities that are studying things that we don't die of and that cause countries around the world to be suspicious of our intent, whether that really is serving our best interest, both in terms of keeping Americans healthy and in terms of global security, because what we do is followed, and there's a tremendous potential, if we don't pull back, to entering into a international sort of downward spiral in terms of this research. So I'll stop there.